All right. So, we are now starting this lecture, and welcome to everybody, including the studio audience. A couple more studio audience now coming in. And my name is Jeremy Smith, and I'm director of the Center for Molecular Biophysics here at the University of Tennessee, Oak Ridge National Lab. And we specialize in biological systems, in particular merging using neutron scattering and large-scale computer simulations to solve problems in energy, environment, and health. So I've got two lectures to give on biological applications of neutron scattering, and they will be fairly free-ranging lectures. This is always what one says when one hasn't prepared much. But before I do that, short introduction as to where I come from. I'm English and studied biophysics as a first degree in Leeds in England. That was the only university that did that. Then did a PhD in neutron scattering at the Institut Laurent-Langevin in Grenoble in France. And then a postdoc with Martin Karplus in Harvard. He won the Nobel Prize a couple of weeks ago in chemistry. And then I set up a group in the French ORNL. The French ORNL is Saclay, the Center for Atomic Studies at Saclay. And after that, moved to see, I'm a bit of a nomad, a wanderer, scientific wanderer, because I set a lab up in Heidelberg after that, the University of Heidelberg, and then came to Oak Ridge 7 years ago. So, all right, so what are we going to talk about? Biology is complicated. The other systems you've been studying are really simple and easy. But biological systems are difficult. Look at this. Oops, let's get this arrow right. This is a complicated biological system. It's cellulosic biomass. What's that? That's plant cell walls. And those are the things that are used to produce bioenergy from cellulose. This is a cellulose fiber. This is the wonderful enzyme called the cellulase that hydrolyzes cellulose into sugars. So there's a chemical reaction going on there. And this stuff is lignin. It's a structural polymer that gets in the way of cellulases from working. So these complicated biological systems, what can neutrons do to help us understand them? Well, how can we understand enzymes working? How can we understand the structures of fibers? So I have a warning up here saying all named hosts have left the meeting. Okay, that means I see, that's no problem. So what can we do in particular to help solve the basic scientific problem, which is how do we efficiently hydrolyze cellulose into sugars, which are then fermented into gasoline or alcohol. So that's the kind of problem that requires a multi-pronged approach, but through which neutrons can help us for some detail. And I'll come back to that later. Remember, we have enzymes, fibers, polymers, and in there, of course, ubiquitously important is water. What I'm first going to do is just review some principles of neutron diffraction here. We'll largely be talking about dynamics, but I just want for the students listening in, for them to be aware of basic techniques in neutron science that are of use for biological systems. Firstly, neutrons, as many people have already told you, scatter from nuclei, whereas X-rays scatter from electrons. So this has several consequences. One of these is that, well, we develop force fields. We want to simulate biological systems. And to do that, we need an accurate description of the structures of the systems that we're looking at. And the structure involves what? Distances between nuclei, for example, a bond length. CH bonds are all over biological polymers, for example. And they have a certain bond length that's defined as the distance between the C nucleus and the H nucleus. Now, neutron diffraction can accurately measure that from crystallography. X-rays cannot. X-rays scatter from electrons. So what you will get there is the distribution in a CH bond of an electron density. That comes out of the Fourier transform of the scattering pattern. So, because the electron density is not centered exactly where the nucleus is, one can get a picture of how the electron density is distorted around an atom. In other words, the atomic dipole moments that allow us to more accurately model using a computer what these bonds look like and how they interact. Another aspect that neutron diffraction is useful for is in the second point here. You see I have points 1, 2, 3 and then 2 points 4. Huh? But here the scattering power does not depend on the atomic number, and hydrogens can scatter strongly, especially incoherent scattering. You've heard of that already. And the deuterium and hydrogen have opposite signs of the scattering length density. So we can use that. We can use that to, for example, in small angle scattering. You've probably heard already lectures on small angle scattering that mention contrast matching, where one can match out one of the components in a small angle scattering profile. This was used in classical work in biology in the 1970s. People knew that virus particles had nucleic acid and protein, but they didn't know where the nucleic acid was, and they didn't know where the protein was. And so to use contrast matching allowed them to figure out that where the nucleic acid was in the middle of the virus surrounded by a capsid, viral proteins. So that was done with that. And nowadays you can use contrast matching to very well in protein nucleic acid complexes, but also in other aspects, other types of biopolymer. Another diffraction technique that's useful for biological systems is solution scattering. This is high resolution scattering, meaning we're looking at atomic pair correlation functions. Solution scattering of, say, amino acid residues in solution or peptides. It can give detailed information on conformations that the peptides have, but in particular on the hydration. Why do we want to know that? Well, a peptide, whether it folds or interacts with another peptide, or whether it aggregates with itself, that depends in great detail, to a great extent, on how it interacts with water as well. The water is critical in biology for forming structures, and solution scattering with isotope substitution allows us to derive so-called partial structure factors, which are Fourier transforms of specific atom pair correlation functions, not just the average over all of them, which is what you get with X-rays. So this allows us to get detailed structure of interactions in solutions. So here's some those are different diffraction techniques. Enzyme structures are very important for us as well, though. I showed you a picture of an enzyme in the previous slide. Here it is. The question is, at an active site of such an enzyme, how does the enzyme catalyze the reaction? How does the enzyme lower the energy of the transition state for the chemical reaction as it catalyzes relative to an aqueous solution? Now, one of the things our lab does is we calculate enzyme reaction mechanisms. So out of different possibilities for a reaction to take place, which is the one the enzyme actually uses? And our big problem in doing these calculations is the hydrogen atoms. We don't know where they're positioned in an active site, and they could jump almost anywhere. So to know where they are is very important. And neutrons, because they see hydrogens, can be used to position them in an active site, and therefore drastically narrow down the number of combinations of reactions that is theoretically possible. So we also have the fact that water structure, in particular hydrogen atoms, can be located with neutrons. You can't see them so easily with X-rays because X-rays scatter from electrons and hydrogen has only got one electron, far fewer than other elements. However, with neutrons, I don't know whether you've had this yet, but the big problem with neutron crystallography is we have no solution to the phase problem in the absence of an X-ray structure. So you need an X-ray crystallographic structure in order to seed the solution of your neutron structure. And also we need relatively large crystals, and of course one cannot buy a neutron source for the lab, unlike an X-ray source. You need something, either a nuclear reactor or, as we have here at Oak Ridge, a spallation source. We've got both of these actually. Yeah. So that's an overview of diffraction. I next want to talk about some principles of neutron scattering. You've seen this before again. This is our scattering vector triangle, in which Q is the scattering vector. And it's a function of energy and momentum transfer. And as I mentioned, you've seen this, but I think it's important. It's always useful to get personal views from different people as to how they interpret this kind of thing. Here's the way I see it. I see it that neutron and X-ray scattering all depends on one function. And that's this function called G of R and T. And that is the Van Hove correlation function. What is it? Well, it's the probability that if a particle is at position 0 at time 0, then it or a different particle will be at a position R at time t later. Okay? So it's a very simple probability function, a very basic function of structure and dynamics. And all you have to do is Fourier transform it, and you get what one measures in neutron scattering and X-ray scattering, in fact. What modulates this with X-rays is these scattering factors that come in front. the form factors, which are not in this equation here. So you see, neutron scattering is a very fundamental probe of structure and dynamics of R and T, and that's my view of it. Okay. You will have seen, presumably, descriptions of the Fourier transform in space of this side of the equation, which describe a correlation function in time. The intermediate scattering function, Fourier transforming that, then in time gives you the what you measure. This is the dynamic structure factor. And this didn't come out very well, but the intermediate scattering function can be expressed in terms of the positions of the atoms. So we have different forms of scattering with X-rays. It's difficult to get the energy resolution here, this omega resolution, so it's effectively equivalent to performing the integral over energy here to get an S of Q for X-rays. And then one has coherent and incoherent neutron scattering that you've heard about. Coherent scattering, the way I look at that, is simply this Van Hoff function. It's when it's the same or a different atom. It can be a different atom there. You can have pair time correlation functions in coherent scattering. Whereas for incoherent scattering, it's the same atom. This is an S down here, G sub S. So it's a self-correlation function. It's where an atom is at time 0 compared to where it will be at time t later. And that's the information you need to plug in to dynamic scattering of neutrons. So where do we get this function from, the G of R and T? Well, you can use theoretical models, simple ones of things oscillating or jumping or diffusing, or you can use a computer simulation. From a computer simulation, the beauty of it is one simulation, because of the weak probe that a neutron is, a simulation can be used to calculate all these quantities. The incoherent scattering, the coherent scattering, X-ray scattering, neutron scattering, and everything. From that, then, you can interpret the results. So, coherent scattering mentioned that. You integrate over time and you get the typical diffraction patterns there. If you don't integrate over time, one can get pair correlations and incoherent scattering. Here's a typical description of a dynamic scattering pattern. Dynamic neutron scattering now. There can be an elastic component, a quasi-elastic component, and an inelastic component. Basically, the way to think about this is if nothing is moving, if the atoms are completely still, all you have is elastic scattering. Then, so that's, say, at 0 kelvins with a classical system. If you then start to heat the system up, then vibrations will appear. Now, this correlation function here will, if there's a vibration happening, there will be a sine wave. There will be a sine wave in the correlation function. You Fourier transform that and you get a peak at non-zero frequency. So this peak is due to some vibration happening. So these are different vibrations then. The peak may have a width. That's due to the resolution of the instrument not being perfect. And if the resolution is perfect, then the peak is due to anharmonicity in the vibration. The fact that my sine wave in the correlation function will peter out to 0 because the vibration is anharmonic or damped. Yeah? So if I keep going, heating up, maybe then there'll be some activated process happen. in which the system jumps over a barrier from one potential well to another one, and that will lead to quasi-elastic scattering. That's stochastic events, sort of random jumping from one place to another or diffusing. And that can be biologically very important, but it'll only happen once the energy barriers have been crossed that allow that to be, these motions to be excited. And each time that this scattering happens, each time that a vibration is excited or a jump, then neutrons are taken out of the elastic scattering and moved into this inelastic, quasi-elastic scattering. It's kind of a conservation law here which says the number of neutrons scattered is always the same. So there's a balance. So that which is taken out of the elastic scattering is put back into the inelastic. Okay. So there's my introduction. Let's move on to computer simulation now. How do we do that? Well, in the end, simulation is a cop-out. It's a pity we have to do them, simulations. It would be much nicer if we could just use mathematics. Mathematics is simple, easy to interpret, but this recognizes the fact that these systems that we want to understand are complex. There are different types of atoms involved, and each of those interacts with different types of interaction. So we'll call that a model system at atomic detail, each atom explicitly represented, and we will then derive a potential function. I don't want to go into that in too much detail. I think the next slide might. But this potential function gives us the potential energy as a function of all the relative distances of atoms in the system. So it's a many-dimensional potential function, and in these many dimensions, one can define an energy surface here, with minima, and barriers, and mountain ranges, and cols, and passes. And exploring that is what we do with computer simulation. Averaging over what we do allows us to appreciate the neutron scattering from the system. So, this describes what we need to do a computer simulation of atomic dynamics, set of atoms, solvent explicitly or implicitly represented, some kind of boundary conditions. I won't go into these details because time is already our enemy. And this allows us then to run a simulation. Here's the potential function, and we'll get back to this. This is a bond with the preferred distance, L0, and we can represent it as approximately harmonic. So we sum over all the bonds, and we have the bond stretching potential. Same with angles. Bending the angles are roughly harmonic, so we assume they are. And you see the parameters, force constants and lengths that we need to get from experiment or high-level quantum chemical calculation. Torsions is another one. This is a dihedral angle between four atoms. And these torsions are chemical. Each of these terms depends on the chemical element, the type of atom, the bonding structure that we're thinking about. And then we have these important non-bonded terms. The van der Waals interaction, which gives us a hard repulsion and a softer attraction, and electrostatic interactions, which occur because the charge on the nucleus and the charge on the atoms don't cancel each other out. There's a net partial charge on each atom in the system for most systems. So that's our potential function. Now, what about neutrons? Let's get back to it. First thing we can do is imagine our system heating up from 0 kelvins that this is an enzyme. As I mentioned, the first thing that happens when you heat something up is vibrations get excited. Now, some of these are high frequency vibrations, relatively high frequencies. And so here's an example for a protein molecule called staphylococcal nuclease of vibrational inelastic neutron scattering, experimental peaks and calculated peaks. So you see there's this big one here in the experiment, and some other ones that become, and this other big one here. Now this is incoherent neutron scattering, so it's mostly hydrogens that are doing the scattering, and these peaks, this one here for example, is a specific type of vibration, and that's a methyl vibration. So the methyl groups in the protein, I'm kind of illustrating with my hand now, they're threefold symmetric. And these are not rotations of the methyl group between threefold symmetric wells. These are librations within the potential wells, the vibrations. So that's a strong vibration. And then there are other peaks too. This particular spectrum was taken maybe 15 years ago. Today spectrometers have better resolution than this one. And why are we interested in this anyway? For those of you that know a bit about molecular spectroscopy, you'll know that we can obtain high resolution infrared and Raman spectra, which give very detailed descriptions of vibrations going on in molecules. Well, the interest for neutrons is, firstly, we see the hydrogens, but also this. The dynamic structure factor, that's the measured intensity, is simply proportional to the square of the amplitude. That's all you need to know. And so, because I mentioned this Van Hove correlation factor was a very direct connection between neutron scattering and dynamics, this directness is manifested in the fact that amplitudes are very easy to calculate. Once you know the dynamics, you can calculate the amplitude of the vibration peak easily. This is not true for infrared scattering, where you need to know dipole moment fluctuations. And for Raman, it's even worse. You need to know polarizability changes. So, vibrational and elastic neutron scattering is complementary to other forms of spectroscopy, has certain advantages that can be used to study these vibrations in complex molecules in some detail. Let's move on now to something of practical use. That's ligand binding. Now, we know in biology that we have big molecules and small molecules. The small molecules often bind to the big molecules. And how this happens, why this happens, when this happens, and the forces behind it are of fundamental interest to biologists. It's of interest because that's how biology works. A ligand binds to a protein. Protein can change its shape then, and a cascade is initiated with some complex biological function, such as some proton pump firing or synthesis or something. But also ligands binding to proteins, those are drugs. Most drugs are small molecules that have been designed to block, inhibit, or affect the action of a protein. So we need to understand that. I'll give you an example of how neutrons can help to understand the thermodynamics of ligand binding now. And that's this fact. Now, when a ligand is unbound, it has its internal vibrations, and it also translates and it rotates. So you have 6 degrees of freedom there. Now when it forms the complex, those translational and rotational degrees of freedom are lost and become vibrations in the complex. You also form new interactions, and this modifies the matrix of vibrational force constants, modifying the vibrational spectrum. So in other words, what I'm saying is the vibrations of the protein change when the ligand binds. Now, structural biologists who've been studying this, they know that vibrations change, but there's been no way to probe that. So they've tended to talk about other things instead, like, do hydrogen bonds form? Or is there a hydrophobic penalty to pay for dissociating a ligand? These things are more easily quantifiable, and so people have been using those in functions to describe protein-ligand interactions. Now the vibrational changes, though, how do we get at that? Well, here's the equation that one uses with neutron scattering to get this. I won't go into it in detail, except for one conceptual thing. Firstly, what we get out is something called the vibrational density of states. And that's just the probability distribution of the vibrational frequencies. How many frequencies there are how many vibrations per unit frequency. Now this can be derived from what we measure, which is the dynamic structure factor. So this is what we want, the frequency distributions, dynamic structure factor. If you remember, I said in the previous slide that this is proportional to the square amplitude. So we need to remove the amplitude information. And that's what this term does. It effectively removes the amplitude by extrapolating to Q tends to 0 and with these factors. One has then the density of states. Okay. Only neutrons can give you this. There's no other technique to do it. So it's fundamental information. And let's see some results now on binding a cancer drug, Methotrexate, to its target, which is dihydrofolate reductase. So this is a simple version of what I just said. What happens now? I understand there are physicists listening, and if so, physicists might like to try and guess what happens. If you bind a small molecule to a protein, will this soften the protein vibrations or will it harden them? Will the protein become softer or harder? You can imagine it going both ways. If you were to soften the protein, then the vibrational frequencies will decrease. The lower the frequency of the vibration, the weaker the effective force constant, the softer the protein is. This also increases the entropy of the vibration. Whereas if you harden the protein, then the frequencies go up. If the frequencies go up, you are stiffening the potential, the effective potential, and increasing the force constant. So here's what happens. This is the complexed protein, here's the uncomplexed one. Now, what you'll see is compared to the previous slide, here, these are frequencies. These frequencies are given in wave numbers. That's the kind of chemist's frequency unit. For those that work in milli-electron volts, 1 milli-electron volt, I believe, is 8 wave numbers. And you'll see back here But these are relatively high numbers, like 1,000 or hundreds of wave numbers. Now, the change in the spectrum on the drug binding is at much lower frequencies. See, that's of the order of 10 wave numbers. So these are lower frequency vibrations, so they are large amplitude collective vibrations distributed over the protein. So if you think of the whole protein with hundreds of atoms moving in a correlated way, those are the vibrations affected here. And what's happening is it's softening the protein. How do I know that? Well, one has a shift to low frequencies, an increase in the absolute number of vibrational modes in the low frequency region. It's been shifted from the high frequencies to the low frequencies. Now, why is this biologically important? Well, if you know this density of states, you know the complete vibrational thermodynamics of the system, and so this can be calculated, the change in the free energy, delta G, and the entropic and enthalpic components of those can be simply calculated from the shift in vibrational frequencies. That was done here. We find a 4 kilocalorie per mole contribution to the binding from the vibrations. This favors binding of the ligand. And it's entropy driven. If you remember, I said if you shift to low frequencies, the entropy goes up. And so there's a partial compensation from enthalpy you see here, but not complete. So a 4 kilocalorie per mole effect. Now is that important? Well, it's a factor of 10 to the 3 in the binding constant. That means that 1,000 times more molecules bind due to this vibration happening than they would otherwise. So it's important. Without this, the cancer drug wouldn't work. So there's some fundamental information. Now the next question is, why does this happen? Why does the protein soften? Many people might have thought it would stiffen when the ligand binds. In fact, it softens. So for that, we need simulation. By the way, this reference gets you into the series of papers that describes all this. If you're interested, the initial paper was in Physical Review Letters, but there have been 2 or 3 since then. And here you see that same experiment with the shift. At higher frequencies, you don't see it so much. And then simulation reproduces the same shift. In other words, you get the same change in frequencies from dynamical simulation as you do in experiment. So that's useful, but it doesn't tell you anything. Confirms the experiment, if you like, but it doesn't tell you the origin. So what we have to do is we have to decompose the simulation. We have to analyze the simulation to find out what's happening. So I'm going to show a couple of results. Firstly, that there's a breathing component, a kind of radial component in the protein. Here's the protein. And there's breathing modes that tend to be affected. That's what's shown here. But also the physical origin. Briefly discuss that without going into any detail. Here's the reference to it. Why does the protein soften? Well, the simple ball and spring model here, you just look at this one. This is what happens when you attach a ligand to a protein and you then create new vibrations in the system. This one. And this new vibration was the external motion before. So adding these new vibrations can be of low frequency, In other words, thermodynamically very important here. This is the entropy that we just saw as a function of frequency of a vibration. It's low frequency vibrations. If the coupling goes into low frequencies, you can have a strong thermodynamic effect. And that's what was done here. This is a calculation of the coupling constant of the ligand external motion from the simulation. And that shows that it's these low frequencies that couple strongly, and that one gains entropy from coupling between the protein and ligand external motion. And this entropy gain is what drives the overall entropy increase and softening of the system on the ligand binding. Okay, so that was a rather complicated description, so I've kind of rushed over. To get the details of it, do read this paper by Kei Moritsugu, who was a postdoc of mine and is now a staff scientist in the Riken lab in Tokyo. Okay, so we're going to move on to even bigger vibrations now. And these are vibrations concerned with protein interactions. If there are any questions, please let me know, including from our studio audience here. Anything people don't understand. Here are two interacting proteins, and you can think of those as balls on a spring as well. You can do that. How do we measure interacting proteins? Well, one unique technique that neutrons possess is the ability to measure lattice waves going through crystals. Here's how we think of lattice waves. If you're a biologist and you're wondering what a lattice wave is, if you have two atoms, say here and here, that are in identical positions, or rather here and here, identical positions, but in different unit cells in a crystal, so they have exactly the same environment, then they must execute the same motions because they have the same environment. But they don't have to execute the motions in phase. They won't. One of them could be going, I'm going to try and point at the camera, up when the other one's going down. Yeah? And that creates a wave that goes through the crystal that's a lattice wave, and there will be a phase relationship given by the wavelength of those waves. That's the phase relationship that you can translate into a wave vector. And of course the vibration has a frequency. When you plot these, it turns out if you plot Q, the wave vector, against the frequency, in a crystal it turns out when you do the math that these vibrations fall on branches. This is many, many vibrations. Follow lines in the space of Q and omega. So these are called phonon dispersion relations. We have acoustic phonons, optical phonons, and physicists will know this stuff, but this will be very new for biologists. With neutrons, you can use a certain type of spectrometer to go into reciprocal space, position yourself at a Bragg peak, and move a little bit away from it. Here, this is small q. That small q, that's your phase relationship. And then measure the frequency. Do an energy scan at this q point. And that gives you then the dispersion relations. Now, this is a standard technique for neutrons in small molecules. It has never, to my knowledge, been really successfully done for a biological system, but people are beginning to try now. They're beginning to succeed. There's some hint in recent papers of this kind of thing existing. I want to show you an example for an amino acid. Here's L-alanine. This is an experimental dispersion relations for this, and one's calculated then from one of these dynamical simulations. So you can do the same calculation in an experiment, compare them, see where they're different. Where they're different, one can try and maybe modify terms in the energy function to give a better representation. Yeah? So that's inelastic. It's called triple axis coherent inelastic neutron scattering. It's very powerful for looking at correlated motions, collective motions between molecules in a crystal. You can even, you remember I said before, amplitudes are easy to calculate. It's just squared amplitudes, and that's basically what's given here. Again, this strange symbol here is a result of Adobe Acrobat or whatever it is. And here you see calculated amplitudes and experimentally measured amplitudes for these phonons that are going through this amino acid crystal. It's useful for understanding the physics present. Now, what about a protein? Well, here I issue a challenge to all experimental neutron scatterers. Do this if you can. Nobody's done it yet because we've calculated it. We've calculated the phonon dispersion relations for a protein crystal. You need a big computer to do it, but we have big computers here. So we calculated it. And there they are. There's acoustic phonons that are longitudinal and transverse, optical ones too. This gives a complete description in the harmonic approximations to the potential. So if you assume the potential is vibrational, say at low temperatures, a complete description of all the motions between the protein molecules in the crystal. And it's there. If you like, it's a physical benchmark for this system. But nobody's been able to measure it yet. Now, why is that? Why has no one been able to measure this? Well, maybe because proteins are big. And if proteins are big, then their unit cells are big. And if their unit cells are big, then their unit cell in reciprocal space is small. And this is a unit cell in reciprocal space. It's called the first Brillouin zone. And therefore, to concentrate a neutron beam in there and measure it is a technical challenge. So that's one reason. But also maybe because there are lots of vibrations going on that might be confusing to measure. But anyway, it's a challenge. And if any of you graduate students listening in Akron or wherever wish to take up that challenge and measure the first ever dispersion relations in a protein crystal, that will be a big deal. Okay, I'm going to probably skip over these, and I want to, not too sure where I am now. Oh yes, molecular dynamics. We've talked about vibrations so far. We've only talked about vibrations in the main. And but there's much more than vibrations goes on in biological systems. There's anharmonic dynamics. The vibrations can be damped, or one can have purely diffusive motion. That's overdamped motion. Or one can have jumps between potential wells. And all these are interesting models of what goes on in a protein system. So, what do we do with our potential function? Well, the molecular dynamics method involves calculating the forces by differentiating the potential energy and then applying Newton's law of motion to calculate the accelerations effectively. One then builds up trajectories from which one calculates the correlation functions that are needed. So here's how we do a typical MD molecular dynamics simulation. take initial coordinates for our system, which have to come from X-ray scattering results, crystallography, in the main. This is a protocol for performing a simulation. Different people have different protocols. Energy minimize maybe the structure to remove forces. Gradually heat it up. Equilibrate the system by taking out temperature until one has an equilibrated potential energy, and then production dynamics over which one calculates the quantities one is interested in. That's typically what one does. And this I will skip over. It's a general description of how one takes a molecular simulation and uses techniques of statistical mechanics to calculate some macroscopic observable. Won't go over that in any detail, except to mention that of the observable one needs to take an ensemble average. And in an experiment, this is an average over the molecules that one's observing and over time. So in a simulation where we only have one protein, then one assumes that the time average is the same as the ensemble average. So one's assuming that the system is ergodic in that case. And it's a reasonable assumption for most of the systems that we're looking at, the ergodic hypothesis. Assume it, and therefore from the behavior of one molecule over a long time, we say that's equivalent to the behavior of lots of molecules in the experiment. We do that, calculate the correlation functions that I showed you before from the simulation, and then you can calculate your scattering densities, your scattering profiles. So there are efficient software to do that. The software that we wrote is called SASIM, and it parallelizes very well on modern-day supercomputers. And you need supercomputers to do calculations of these things from large simulations these days. From small simulations, it's not so important, but from large ones on large systems over long times, you need high-performance computing. All right. So some more details on simulations. There are different timescales of motions involved. I've alluded to that before. 10 to the minus 15 seconds is the fastest type of motion in any molecular system. Collective vibrations, such as those that get modified on ligand binding that I showed you, those are about 1,000 times slower, picoseconds. And then we have interesting conformational transitions in biological systems. Side chains jumping from one position to another, loops closing, hinges bending, things like that. And they can be much longer in timescales. They range from picoseconds through to any time. And the motions involved in catalysis, directly involved in the catalytic reaction, those can be microseconds or milliseconds. As mentioned here, just as an aside, yeah? I mentioned the Nobel Prize in chemistry of this year was given to computational chemists. One of them who won it was Aryabhashal. He's from the University of Southern California. And over the last 10 years or so, Varshal has been rather preoccupied with the notion that dynamics plays no role in catalysis. Motions of enzymes have no effect on the catalytic power of enzymes. There are other people that say motions are important for catalysis. Now, how could that be? Well, it's clear that enzymes must move to function. If you froze the atoms in place, you would never get any reaction to take place, other than electron transfer. So it's not that motion is not necessary, it's just whether dynamical differences of a protein in solution compared to in the enzyme active site, differences in the dynamics contribute to the catalytic effect. In other words, the increase of rate of the reaction in the enzyme relative to in aqueous solution. And that's a disputed point right now. So we've also mentioned how dynamics affects ligand binding and protein folding. Folding of a protein up is itself a dynamical process, of course, not one that's in the range of neutron scattering, at least not for now, but it's dynamics in itself. What about MD? Well, we need to set our integration time step to be that of the fastest varying force. Otherwise, the whole thing explodes. We don't integrate finally enough. And that's a femtosecond. That limits us. That limits us. The accessible timescale now, well, I've put about 100 nanoseconds in this side. It's now a microsecond. It's fairly routinely accessible with specialized computers like the one that D.E. Shaw built for hundreds of millions of dollars. So he's built a kind of computer SNS, if you like, cost about the same, and it allows us to now simulate to milliseconds, so 1,000 times faster than the timescales that are commonly accessible. So we'll move on now to anharmonic effects. I think there are about 5 minutes. In fact, I'll just introduce that. What I do want to know is that if anybody has any questions, please type them, because question answering time would be now. Question answering time would be now. And if it isn't, I'll go on for about another 5 or 10 minutes, and we will end. I want to talk about what happens when you heat a protein up. Here's the elastic scattering. The elastic scattering is S of Q, that's the wave vector, and 0, meaning zero energy transfer. And to a reasonable approximation, it's given by this function. It's exponential to the minus Q squared delta R squared. So, over the H atoms. This is for incoherent scattering. Elastic incoherent scattering. If you remember, I said that if nothing moves, then what we have is this is 0, and the elastic scattering is 1. In other words, the elastic scattering is a maximum. As soon as things start to move here, then this exponential to the minus x kicks in, and we start to get a decrease in the elastic scattering. So, by going to low Q, this is where this is valid, and plotting S against Q squared, or log S against Q squared, one can derive this. This is the mean square displacement. It's the spectroscopic mean square displacement. What that means is, going back to the Van Hoff function, it's where an atom is compared to where it was, and that's different to a diffraction mean squared displacement. Crystallography, that's X-ray crystallography or neutron crystallography, gives us fluctuations from the mean. Spectroscopy gives us time correlations. There's a difference there. Henry Glide knows all about this, by the way. We have a postdoc here from Henry Glide's group in Delaware. Oh look, we've got some questions. The question is from Ming, can we calculate the energy state with the phonon dispersion relationship of the protein? I'll repeat that question. Can we calculate the energy state with the phonon dispersion relationship of the protein? So, depends what we mean by energy state. What the phonon dispersion relationships give is the entire dynamics of the inter-protein motion. So given the harmonic approximation, in other words, assuming that there are just vibrations, then you have a complete description of the energetics of the inter-protein, the protein-protein motion. And in fact, these calculations can be generalized, and we have to include the internal motions too, even though the intra-protein motions. And in that case, yes, it's a complete description of the energy state of the protein. It's a complete description, both internal and external, but assuming that there are only vibrations. That's the answer to that. Penn State question. Would you please remind me why we only look at the dispersion relation within the first Brillouin zone? And the answer to that, I believe in the second and third it's the same, but the first Brillouin zone is experimentally easily more accessible. Penn State, why are the error bars at high frequency, why are they larger? So I'm wondering which slide we're referring to now. Let's see if I can get back to error bars at high frequencies, because that might be in the dispersion relations. Which one? Slide number 9. Where do I see slide 9? That's the oldest. I see 16. That we're going back to slide 9. Aha. That is because the this function. I'll go back here. The measured intensity is proportional to the square of the amplitude. Now the statistics is proportional to the intensity. In other words, the higher the intensity of the peak, the better the statistics you get. Now, one equation that's not on here is that the squared amplitude is proportional to 1 over the squared frequency. So low frequency vibrations have higher amplitudes. You can see that here actually. This is the dynamic structure factor, and these low frequency vibrations, they have high amplitudes. This is your 1 over omega squared variation coming in here. And that is why the statistics are better at lower frequencies. Because the amplitude here, you can't see, this is the density of states, so it's had the amplitude removed from it. But that doesn't remove the amplitude from the statistics. So Min, again from Penn State, is saying, so that doesn't consider the conformation of the protein. That's the question. So that doesn't consider the conformation of the protein. The answer is the vibrations all take place in one conformation. A vibrational analysis. However, what will happen at low temperatures is the proteins will be in, each different protein will be in a different energy state, a different conformation, in other words. Many different conformations. Each of these conformations has its own vibrational spectrum, and these will be averaged to produce what one sees experimentally, and ensemble averaged. See, so there's an average over the conformations of the vibrations in each conformation. Alright, so I see here that Ming is typing, which means Ming has another question, I guess. When that arrives, we'll come to it. I think we will, Penn State say thank you. Thank you very much, Penn State. We'll move on to slide 23 again. Just describe briefly what this is. This is a measurement of the elastic scattering of a protein, myoglobin. Myoglobin is a protein that transports oxygen between the lungs and the muscles. So, it's the first protein for which we knew dynamics was important for function. And that came from the crystal structure of myoglobin. Max Perutz, the great crystallographer, saw there was no way that the oxygen that myoglobin binds could reach the iron atom that it binds to without the protein moving out of the way, so to speak. Yeah? So it's always been studied. And what you can measure with neutrons is an average over the hydrogen atoms of the mean square displacement. You get that from this elastic scattering. So at 0 kelvins, this mean squared displacement, I'll come back to Ming's question in a minute. Yeah? 0 kelvins, mean squared displacement is 0. 50 kelvins is very low, and you see that there's approximately a linear increase at low frequency. So this is consistent with harmonic motion. There only being vibrations. And if this were to continue, you only have vibrations in the protein, then this straight line would be what one observes. However, what is seen with the neutron spectroscopy is that extra motions occur. The mean squared displacement is higher than that would be given from harmonic motion. So there's some kind of transition here. And this transition resembles that between gases and liquids. The other thing that's been interesting is that in some cases at least, proteins start functioning at this temperature roughly where one sees the onset of the transition from a glassy to a liquid state. Now, let's think about this glassy state, and in doing that, one can answer Ming's question, which I'll repeat in a minute. You've got to consider the energy landscape of a protein. It's got many different minima. Each of these minima is a separate structure of the protein. They all have the same overall fold, but they're distinct conformations. And when I'm at low temperatures, the protein is frozen in these conformations. It cannot jump between one and another. So Ming's question. Ming says, is it possible to calculate all protein conformations to get the energy population of one protein? Well, we don't know for sure what all the conformational states at low temperature are, but we can try and calculate them and average over them, and that has been done by us and others. So in each one of these states there's a vibrational spectrum, for example. Calculating each of these states, averaging over them, well, we're doing that now with something called a meta-stability analysis of protein conformations. Maybe I'll have a chance to talk about that in the next lecture. Okay, so just about at the end, there's one more question coming from Ming. And in the next lecture, we'll describe this protein glass transition in more detail. Ming's question is the measurement done in protein solution? It's a practical question. And the answer is sometimes it's done in solution, but then you have to be aware of the scattering from the solvent and the overall rotational and translational motion of the protein molecule. The protein global motion, the whole molecule translation and rotation, are on similar timescales to the conformational transitions. Distinguishing between them using neutron scattering is important. You don't have that if the measurement is done in a powder form. So many people do experiments in hydrated powders. This is, if you like, lifelike powder conditions in which the protein can still function, etc., but doesn't translate and rotate. So that way one sees only internal motions. Okay, so I'm wrapping up already. I've introduced the glass transition. Just summarize what we talked about today. We reviewed some diffraction methods very quickly, just to make sure you're aware of diffraction in biological systems. We talked about vibrations, high-frequency vibrations that complement Raman and infrared scattering, infrared spectroscopy, and we talked about theoretical methods, computational methods to calculate dynamics. We talked about low-frequency vibrations, how they're modified on ligand binding, and we talked also about incipient glass-to-liquid transitions as you increase the temperature of the protein. We'll talk more about that next time. So Ming is typing one more question. If anybody else has a question, I'll answer them. Otherwise, I will end. I'll wait for Ming's typing to come through. One conceptual question: what's the difference between diffraction and scattering? Scattering is a more general term in which there is not necessarily a phase relationship between scattering objects. Diffraction is what happens from a crystal when all the atoms are lined up, and you therefore get scattering waves that reinforce each other and lead to peaks. Incoherent scattering of neutrons is not diffraction because there is no, I'm talking about coherent in the sense that waves will interfere positively with each other in the diffraction pattern. That's what you need for something to diffract. And so with that, we'll end the lecture. If you have questions, next time I'm giving another lecture, right? So if you have questions ready for the beginning of the next lecture, I can answer those. And until then,